So it is my pleasure to introduce our next speaker, who is also my PhD advisor and mentor, Dr. Susan Murphy. Dr. Murphy is a tenured associate professor and chief of the Division of Reproductive Sciences in the Duke Department of Obstetrics and Gynecology. She received her PhD from Wake Forest University and completed postdoctoral training at Duke, where she stayed on and joined the OBGYN faculty in 2003. She's devoted the past 22 years to epigenetics research with a particular focus on the developmental origins of health and disease. Dr. Murphy currently leads the multidisciplinary CIPHERS project, which focused on the impact of cannabis use on the sperm epigenome and potential heritability of these effects. Thank you, Dr. Murphy.
Thank you, Rose, for that very kind introduction. And I just want to thank Jill and Rose and Jordan for the opportunity to talk to you today in this format, which has been really exciting. So apologies for the punchy title, full of peas, but I'm going to jump right in. So why is it that dads actually have been rather discounted in the developmental origins of health and disease? Well, it has to do with reprogramming and what was traditionally thought of as a way for dad not to contribute his epigenetic information to the offspring. So in the first round of reprogramming that happens in the primordial germ cells, methylation is completely erased and then reestablished once sex determination happens. The second round of reprogramming happens post-fertilization, where both the maternal and paternal pronuclei are erased of their methylation information almost completely. There are some genes that escape this erasure, including imprinted genes and repetitive elements. And so it was thought that really there was no way that dad's epigenetic information present in sperm could be carried forward into the child other than at these specific regions. However, as already referred to in the last presentation, in 2015, there was a paper published in Cell showing that there are regions, thousands of regions in the genome that are referred to as escapees that actually are able to carry forward information from the gametes into the offspring and survive that post-fertilization round of reprogramming. So I think it's time that we actually do turn our attention to dads and the potential that they are contributing something that's carried forward into the offspring. So with regard to cannabis, the cannabis that is being used today is not the cannabis of the 60s. So the cannabis between 1960 and 1980 contained about 2% or less of THC, tetrahydrocannabinol, which is the main psychoactive component in cannabis. Between 1995 and 2015, there was a 212% increase in the THC content in cannabis. And in 2017, a survey of the dispensaries in Colorado showed that the THC content in some of their popular strains ranged from 17 to 28%. What's even potentially more troubling is a tendency for a lot of individual products that are available in states with legalized recreational cannabis and available elsewhere that have up to 95% THC content. And these are increasingly used as people try and enhance the high that they get from otherwise using just straight up cannabis. So one of the questions, given that the widespread legalization that's occurring with cannabis and the potential for a father to carry forward epigenetic information in his sperm, is whether or not cannabis can induce changes in sperm DNA methylation, which had not been addressed previously. So we conducted a pilot study, and I'm just going to breeze through the results here. We've already published this data in Epigenetics in 2018, but this shows that yes, indeed, there are changes in sperm DNA methylation that are associated with cannabis use. This is a group of 12 users of cannabis and 12 controls. These men donated their semen and underwent semen analysis at the Duke Fertility Center. The results in the heat map show 46 genes that had more than 10 affected CpG sites. And those genes are listed on the right along with the number of sites. I'll just point out two. One is AHRR, which by far had the most number of CpG sites affected with 94. And this is interesting to us because AHRR has been one of the most commonly identified genes to be altered in its methylation status in babies in cord blood that were born to smoking mothers. But I'll point out in this cannabis study, what we're seeing in sperm is a different region than what has been identified to be affected by exposure to maternal smoking. The other gene I'm going to spend a little more time talking about is DLGAP2, which had 17 affected CpG sites. So DLGAP2 is disc large associated protein 2. It's actually hypomethylated at 15 CpG sites and hypermethylated at 2 in the sperm. It's involved in synapse organization and signaling. It is an imprinted gene, but it's imprinted only in testes where it's paternally expressed. It's also expressed in the brain, and it's biallelically expressed in brain tissue. It has been implicated in several studies in autism as quite a strong autism candidate gene, as well as in schizophrenia and I believe in alcohol abuse disorders. This gene also came on the radar for us, not just because of our data, but another study that was published by Albert Rees and Gary Hulse, where they did a survey of the states in the United States that had any form of legalized cannabis and correlated that with rates of autism. And what they found actually was a pretty striking correlation between those states that have recreational cannabis legalization and higher rates of autism diagnosis, and predicted that that was going to be an ongoing effect and likely to get worse as we expand legalization. So of course, we were interested in DLGAP2 for this reason as well. We validated the reduced representation bisulfite sequencing data using quantitative bisulfite pyrosequencing, and these are the results shown here for 10 CpG sites that were contained in the assay designed, where we can see that the control individuals have higher levels of methylation, significantly higher than do the exposed individuals for each one of these sites. We were also interested, though, in what does this mean? So the sites that were identified in this gene were actually intronic, and we're not promoter. We wanted to know if there was any relationship between the methylation of this particular region and the expression of this gene. So in order to look at this, we looked at conceptal brain tissues from 28 individuals from 67 to 122 days gestation. These are the results showing all the individuals, which show a significant inverse relationship between methylation and expression of this gene. But we were also interested in looking at this relationship by sex, since we knew autism was 4 times more likely to be diagnosed in males. Interestingly, actually in males, of which there were about 15, we didn't see a significant relationship, although the trend's still there. But there was a very strong relationship in females that seems to be driving the significance. And this sex-dependent effect is very interesting in the context of autism, and also shows that actually this intronic region that was showing differential methylation in sperm, if that is carried forward to the offspring, could have a detrimental impact potentially on the expression of this gene in the child. To address the potential for intergenerational heritability, that's a challenge to do in humans. So we used a rat cohort. We had ongoing parallel rat studies in which the male rats were exposed to THC at 4 mg per kg per day for 28 days. The sperm were isolated and underwent the same technique, reduced representation bisulfite sequencing. And the CpG sites that we found in humans are not conserved in the rat. So we looked for other regions of this gene that actually were showing any kind of difference in methylation between the exposed and controls, and found a region near the promoter, and that's where these 8 CpG sites came from. So there are significant differences, with the exposed rats having lower methylation at 4 of the CpG sites analyzed. To determine whether any of these effects were potentially heritable, we looked at F1 offspring born from fathers who were exposed, mated to non-exposed females, and found, looking at the hippocampus, there was only 1 CpG site that showed a significant difference, but it was not one of the ones that was observed in the sperm. However, in the nucleus accumbens, which is involved in addiction type phenotypes as well as other things, we did see that the second CpG site showed a significant difference in the same direction, and it was one of the CpG sites that were identified in sperm as being altered by the THC exposure. So this gives us some evidence that there is a potential, at least intergenerational heritability of an effect at this particular gene. Okay, moving on, we were interested because of DLGAP2 in what other genes that are involved in neurodevelopment, what's happening with those in terms of exposure, and do different types of exposure affect the same CpG sites? That's something that has had very little attention. So as you can see from the background picture here, we actually looked at effects of tobacco or nicotine and THC in our rat model. And I should have mentioned, this is work that I'm presenting was done by Rose Schrott, who is one of our moderators, and I'll give her a shout out at the end too. But what Rose did was identify from our reduced representation bisulfite sequencing data of sperm, the 627 named genes showing a significant difference in methylation between groups. She plugged those into String platform, which recognized 574 of those genes, then identified those that had an annotation of neurodevelopment, but importantly also showed more than a 10% difference in methylation. So this narrowed down the list considerably. We wanted to look for something that potentially had a meaningful and potentially strong effect. She took the other 53 genes that weren't recognized by String and did a literature search and found one more gene that actually was involved in neurodevelopment and met this criteria of greater than 10% methylation difference, giving her a total of these 7 genes, which were significantly enriched for social behavior, for glutamatergic synapse function, and also for publications in autism. In the String database, what we were surprised to see was that there was interaction between all of these 7 genes, as well as DLGAP2, the gene I already talked about. And you might say, well, that looks like they were pre-selected. They actually were not. This was a surprise that we found out after the fact. And actually, to confirm this very significant enrichment of interactions amongst these genes, Rose took other groups of 7 genes from the data set, plugged those into String, one by one, testing to see if we could find anything else that looked like this. Was just this a chance finding, or was it something that was real? And there were no other relationships like this that she found with any other 7 genes that she plugged in. So she developed assays to look at these genes in rats that were exposed to THC. This is looking at the sperm. The red arrows indicate where there is significance in the difference between the exposed and unexposed group, with the controls in gray. For 2, there wasn't significance, DLG4 and grid 1. And then moving on to nicotine exposure, these are rats that were exposed to 2 mg per kg per day of nicotine by an osmotic mini pump for 56 days. Again, controls are in gray, the nicotine in red. This is again reduced representation by sulfite sequencing data. And what we see is significance for 4 of the genes, but no significance for 3. What I want to call your attention to is those that are significant in both. These are the same CPG sites that are affected by both THC and nicotine. And what was rather remarkable and surprising to us was that if you paid attention, they're in the opposite direction. So they're significant, but going in completely different directions, which is very interesting, suggesting that these particular genes have some inherent plasticity that responds to specific types of exposures differently, but also suggests there may be vulnerability to more than just the nicotine and THC, and brings up the question about what about mixtures? So for example, a lot of the users that we excluded from our study of cannabis in humans used both nicotine and as well as cannabis, so tobacco and cannabis together. And actually we are planning to do some studies looking at that, because the question would be, do you actually both of them ameliorate the effects of these particular regions? So one of the things that has struck us over and over again in our studies of cord blood and in the sperm is we keep identifying genes that are involved in early development, and in the cannabis studies specifically, pathways in cancer, which is full of tumor suppressor and oncogenes that are proto-oncogenes that are usually involved in very early embryonic growth. And so one of the things that these have in common and that we wanted to look at was the presence of bivalent chromatin. So bivalent chromatin, by analogy, is like being in your car and having your foot completely depressed on the brake and the gas pedal at the same time. So these genes are in a state of repression, but can quickly be activated at the right time and place, and then deactivated again to allow for very specific temporal and spatial expression. The other thing about these genes is that they seem to exist in regions that are retained on histones that have not undergone the histone protamine exchange in sperm. So it's been shown that the retained histones are actually enriched at bivalent promoters in sperm. So what Rose did was take our RRBS data from the human sperm, took the genes that were differentially methylated there, overlapped that with genes that had been identified as having bivalent chromatin, and this was done looking at human embryonic stem cells. So she took the full list of the identified genes and looked at the overlap, and there was a significant overlap between the genes that we identified as being altered from cannabis use in sperm and the bivalent chromatin genes. But then also because of this tendency or the analysis that we've done showing an enrichment of autism-related genes, she also took all of the genes in the Safari Autism database, which has roughly 1,000 different genes that are all highly curated and are annotated as being candidate genes for autism. And there was significant overlap with both of the data sets there as well, as shown by the p-values down here. So this suggests that bivalent chromatin may actually render genes that are characterized by this feature being vulnerable to effects of environmental exposures, and in our case, what we've shown with THC as well as nicotine. So now I want to just finish up by talking about brand new data. We just got this data literally this week. Rose and I have been actively doing some analysis so that I would be able to present some of these results to you. So keep in mind, this is kind of a preliminary analysis. We have made use of an publicly available database to do some of the analysis, and we're going to move that to the ingenuity pathway analysis once we have a little more time. But I wanted to at least start going through some of it to show you. So this is to address the question, which we wanted to do since our pilot study: can the epigenetic effects of cannabis exposure be mitigated by abstinence from use? So this is a human study. We've also done this in rats. Rose is going to talk about that a little bit later. But we asked the men to just say no to cannabis use for 11 weeks, which is, some of you probably recognize, is spans the duration of a spermatogenic cycle in humans. This is a study design. We had 18 cannabis users who completed the study. We had a few who couldn't make it, but they were actually provided a semen specimen before beginning the abstinence period, then went through a 77-day period where they underwent weekly drug testing to make sure that they abstained from cannabis and tobacco use. And then had another semen specimen donated after that abstinence period, which then underwent whole genome bisulfite sequencing. We did the same thing with a non-user group with the 2 semen specimens separated by the 77-day period window, which in the cannabis users was their abstinence period, and did whole genome bisulfite sequencing. We were targeting 60x coverage with this. These are the demographics of the participants, which I'm not going to go over extensively. They really didn't vary. There was a little bit of a tendency for the users to be younger, which is probably not surprising, but it didn't quite reach significance. We also looked at the semen parameters, and there was no significant difference in any of the semen parameters, which actually contrasts with our pilot study where we had a smaller number of individuals, but there we saw a 50% decrease in sperm concentration in the users relative to the non-users, but here we see no significant difference. Although I'll point out that in these parameters, the users do have lower numbers than the non-users. So this is the analysis that we have been able to do so far. First, we took the users versus the controls in the before abstinence samples and identified the top 10,000 differentially methylated CPG sites. Then the users versus the controls in the after abstinence samples, 10,000 significantly different CPG sites, and looked at the overlapping sites from the before and after. This is addressing somewhat the question of whether or not we can ameliorate the effects, but I will point out that for all of these sites, I don't know yet the direction of the change in methylation and whether or not it went one way or another. This is just looking to see whether or not there's any overlap in the significant sites that were identified at both time points. This is a volcano plot that is not meant for you to read all the gene names, but it shows the before group and the after group, the comparisons. And we used a very high threshold for significance with a Bonferroni adjusted significance p-value of about 3 × 10⁻⁹. The solid dots here are those that achieved Bonferroni significance in each case. The lighter colored dots are those that were nominally significant, and then the gray under the line are those that were non-significant. And one thing I'll point out looking at this is that there are a lesser number of Bonferroni corrected labels here for significance in the after, providing some hint that maybe there is some amelioration going on. This is now comparing or looking at the overlapping sites and the KEGG pathways that were enriched with genes in these overlapping sites. And the top ones were glutamatergic synapse and circadian entrainment. The red arrows indicate pathways that we had identified from our reduced representation bisulfite sequencing, the pilot study. And we think that this is encouraging that we're identifying many of the same pathways, and also helps illustrate the benefit of doing whole genome bisulfite sequencing in the ability to identify additional regions that weren't identified in the study with the reduced representation bisulfite sequencing. For comparison, we had about 5 million CPG sites that were analyzed for the reduced representation data, whereas here it's about 17 million. Now, the next analysis is comparing the users versus controls, the before samples, the top 5,000 CPG sites that were differentially methylated, looking at these same sites in the after samples. And here we are addressing amelioration. So what happened to these same sites in the controls afterwards, whether or not they were significant. This data shows the effect of the before versus after for the same CPG sites that were either hypomethylated in the users relative to the controls, and the controls are set to 0 here. So this is showing hypomethylation, and what happens after to those same CPG sites, which are connected by the line. And the dots are representing the mean methylation in those groups. And you can see that there is a tendency for the methylation to increase in the after samples, suggestive of amelioration. Same thing for the hypermethylated, just in a different direction. These are genes or CPG sites that were hypermethylated in the users relative to the controls. They decreased in methylation after the abstinence period. So then looking at amelioration and trying to narrow this down, and we had to pick an arbitrary definition of what we considered amelioration to be, and we can analyze this many different ways, but using a greater than 5% difference in methylation before and a less than 5% difference in methylation after, there were over 4,000 CPG sites that were identified. And of these are now ones that we, by this definition, would consider to be ameliorated or close to being ameliorated. Glutamatergic synapse shows up there. That was the top hit in the overlap. The retrograde endocannabinoid signaling, adherent junction focal adhesion. These red arrows are the ones that showed up in the initial study, just looking at the overlap. So it looks like, and these are all FDR significant, that there is amelioration going on, which is really potentially great news. So of course, one of the other questions is what isn't ameliorated? Now, if you note here, this is just taking the ones now that really show no change, no more than a 5% difference in methylation change. They had a 5%, more than 5% before, and they were still more than 5% afterward. This is now using an unadjusted p-value because there were no pathways that were significant at an FDR level of significance. And there's a set of pathways here that are, that do show no amelioration, and interesting, like estrogen signaling, and there's several that are involved in cancer that are showing up, but we have a lot more work to do to really look into this and see if this holds up. So in conclusion, we've shown that by RRBS and whole genome bisulfite sequencing of completely independent cohorts, we've confirmed that reproductive age males who use cannabis do have altered methylation in sperm relative to the controls. We've shown that DLGAP2, which is an autism candidate gene, shows hypomethylation in sperm with cannabis use, and that the hypomethylation is actually functionally relevant in human brain tissues. And we showed that there's some evidence of intergenerational transmission in rats. Genes that are involved in neurodevelopment at the same CPG site are altered by both THC and nicotine, but some in the opposite directions, indicating some inherent plasticity or vulnerability that seems to be exposure specific. Genes that have bivalent chromatin in sperm seem to be vulnerable to cannabis and THC, and many of these genes are autism candidate genes. And abstaining from cannabis use for the length of a spermatogenic cycle seems to mitigate some of the methylation changes that we're seeing in sperm, but we need validation and independent confirmation in order to, and if we do see that, that would suggest, importantly, and is actionable, that stopping cannabis use prior to attempting to conceive, at least for the duration of a spermatogenic cycle, may lead to improved health of the sperm from an epigenetic standpoint, and hopefully improved chances of having an epigenetically healthy offspring. With that, I want to acknowledge all of the people who've been involved in this study from the start and the funding Templeton Foundation for this. Rose Schrott, as I mentioned, has done the vast majority of the work that I presented here today, but there have been a lot of people behind the scenes involved, as well as several other departments. This really has truly been a multidisciplinary effort and would not have been possible without everyone involved. So thank you for your attention, and if there's time, I'd be happy to take questions.